Okay, so hi everyone. Welcome to lecture 5 of our quantitative genetics lecture or module within our Agri-Informatics lecture series. Lecture 5 will cover single trait selection. And today we have Professor Bruce Walsh with us again. So just to quickly introduce Bruce again, I won't take up too much time. Bruce is a professor of ecology and evolutionary biology at the University of Arizona and an associate head department of ecology and evolutionary biology at the University of Arizona. Bruce Walsh's interests are broadly in using mathematical models to explore the interface of genetics and evolution, with a particular focus on two specific areas: the evolution of genomic structure and the analysis of complex genetic characters, also known as quantitative genetics, which is why he's giving us these awesome few lectures. So, Bruce, without further ado, you are welcome to start sharing your screen and kick off the lecture.
Thank you. Good morning, everyone. Welcome back. Let me help you plan ahead. So today's lecture is going to be right about 45 minutes. Whoops, hang on a second here. Let me get today's lecture is going to be right about 45 minutes. And the lecture tomorrow is going to be running slightly longer. And then the lecture on Wednesday is going to be about 70 minutes. So just plan ahead for you on that. Um, Let me again remind you that, give me a second here, that there is readings associated with each of the lectures. That's background readings. Today's background readings are from the 2018 book. You've got those chapters in your folders. That's chapters 13 and 16. And again, I stress in all these lectures, there's a lot of details, and the important points isn't the details. You can look those up in the notes. The important points are to get some of the basic concepts, the basic ideas down. Once you have those basic ideas down, you can actually look up the details. So focus on the ideas, not the details. You can look up the ideas. Give me a second here. For some reason, my thing is not advanced. Okay. So this lecture is broken into two parts. We're considering selection on a single character. I'm first going to talk about predictable changes in the mean, and this is from chapter 13 called the Breeder's equation. Then I'm going to talk about predictable changes in the variance. So when we think about this response to selection, for example, I want to select plants that are a certain height or a certain root depth or ability to survive a certain fungal pathogen. So what happens with selection is you have a crop or animal that you're growing, you subject it to some sort of criteria. Either nature does that, for example, there's a drought, or you do that as a breeder, or usually it's a combination of both. And what you see is the group of parents you decide to move forward is a subset of the original population. And if you measure the mean in that group of parents, it typically differs for your trait of interest from the mean of the entire population beforehand. And that choice of parents is a within generation change. What you want to then do is ask how much of that within generation change, for example, I have selected the tallest parents, how much of that translates to the next generation? What's the height of their offspring? And that's a response to selection. And as we talked about on Friday, parents pass on breeding values to their offspring, not their phenotypic values. So you only get a response to selection when you change the breeding values. Now remember, the breeding values is simply a parent. You take the alleles in it and you sum up the average effects of alleles for that parent. We don't need to know these. We can estimate the breeding value directly. But the bottom line is that a within generation change is caused by selection. A between generation change, a response to selection, is caused when selection changes the distribution of breeding values. I choose large parents if those large parents also have large breeding values, that is, their breeding values for height or above zero, then their offspring will improve in the population. If when I choose large parents, their breeding values are zero, the population won't improve. And we'll quantify that in a second. So let's measure two metrics. Metric number one is the selection differential. That's the response to select, and the selection differential, S, right down here. That's the within generation change. So again, I have a whole room full of individuals. I pick a small subset of those as parents for the next generation, And the mean of those subset of individuals is mu star. The mean of the entire population is mu, and that difference between the mean of the selected parents and the mean of the overall population is the within generation change, selection differential S. The response to selection R, if I take those parents and mate them at random, what's the mean of their offspring? Well, mu t plus 1 is the mean of the offspring in the next generation. Mu of t is the current mean, that is the mean before selection. That's the change in mean. So what I want to do is connect the within generation change S to the between generation change R. And the way we do that is by what's called the breeder's equation. So let's look sort of pictorially what's going on here. So I have a histogram, and the upper panel is individuals in the population. And then let's say choose the upper 10%. That's truncation selection. That's this little wedge here. I measure the mean of that little wedge, and I look at how that mean differs from the overall mean, and that's the within generation change. That's the mean of the parents, and how much the mean of the parents differs from the overall mean. Then I cross those parents, and I get offspring, and in those offspring, the mean has improved relative to the original mean. And we call that the response. Notice, though, that the mean here, the response is smaller than the parents I choose. Why? Because parents are imprecise predictors of their breeding values. And so in this situation here, I get a large response, a large within generation change, and then I get a smaller response. So what parameter connects these two? And the answer is the heritability. So again, you can look up the details about this, but I can write the regression of an offspring on its parents. So here's the value of the father and the mother. There's the overall mean of the population beforehand. The expected mean of the offspring is simply the deviation of the parents from the mean times the heritability. So if I average over all the parents, well, if I average this equation over all the parents, this is simply mu star. That's the mean of the parents. So this is mu star minus mu. That's S. And this is the mean beforehand. That's the mean of the offspring. So that difference is R. So you get R equals H squared times S. And this expression down here is called the breeder's equation. It was not never formally called that when it was originally appeared, but J. Lush in 1937 proposed that in a series of, at that time, mimeos, which later became a book called Animal Breeding Plans. And this was the first application of quantitative genetics theory to plant or animal breeding. And so this breeder's equation underlies most of how we think about single trait response. And the key issue is the connection is this heritability. Couple of key parts. No matter how strong selection is, no matter how intense I apply selection, my parents are much, much, much bigger on average than a random individual from the population. If heritability is small, the response is small. So that narrow sense heritability, as we talked about last time, the ratio of the additive to total variation, that's a key component. That tells you how easy it is to improve a trait. If heritability is small, 5%, selection response will work, but it's very imprecise. If selection is large, 20 or 30%, selection will respond quite quickly. So S is a measure of selection. R is a measure of response. This is often phrased from an evolution standpoint, is S is the ecology, R is the evolution, and H squared is the genetics. That's how those parameters are kind of connected. Now, this expression of R equals lowercase h squared times S occurs in situations where I take parents and I randomly mate those parents. The other scenario in breeding is I have a whole series of lines that I've replicated. Then I simply pick the best lines. Instead of crossing them, I either clone them or inbreed them among themselves. So in that case now, the response also follows the breeder's equation, but now I've got the broad sense heritability, capital H squared, replacing lower sense H squared. And that's an expression for selection among clones, where again, the distinction is when I choose a clone, I don't cross it with others, I simply keep it in my population. And we'll come back to that in a little bit. The other thing is this very simple equation down here, we can start to decompose and it'll tell us a lot about what we can do as a breeder. So for example, if males and females have different amounts of selection, I can replace S by the average selection on both parents. So I may select more on males than are females, um, or vice versa. And a classic example of this is I go out in the field and I pick those individuals that show the most grain. And I then use those individuals to make the next generation. So in that case, have I selected on the pollen parents? No, I have not because I haven't scored grain on the pollen parents. So in that case, there's selection on the females, but not the males. And now my response is R equals H squared over times S. But now S is S female selection divided by 2. And this is a scenario called pollen control, where I've really only selected on one parent because I've chosen parents based upon the output of the female, ignoring what the trait value in the male was. And this is very common in plant breeding settings. And so how can we account for this? Well, one of the classic approaches is to use one more generation. So take the seed, grow them out, and use pollen from that seed to cross to make the next generation. That results in double the response because I've chosen both parents, but it takes twice as many generations, so the response per generation is the same, and people typically don't do it because of that. Now, I mentioned clones. The idea is imagine we have a room full of people, so all the Zoom people are in a classroom. Normal selection is I'd pick the upper 5%, then I'd randomly mate them among each other and look at their offspring. Under clones, I'd pick the upper 5%. Again, each individual is the average performance of the clone. Those are measured with error. I pick the upper 5% of clones and then use those clones by clonally propagating them to make a new population. So in that case, response is now the broad sense heritability. When you do that, you find a single generation of response is larger than randomly crossing parents. Why? Because broad sense heritability contains additive variation plus all other variation. So remember, heritability is a ratio of some genetic variation over the total phenotypic variation. In narrow sense heritability, it's the additive variation. In broad sense heritability, it's all the variation, including the additive. So with clones, a single generation selection gives you larger response. Now that might sound useful, and it is, but it's not a great solution. Why? Imagine I've got a room and there's 20 different clones. I don't know there are 20 different clones, but there are 20 different clones. Each generation, I pick the top 5, grow them out, repeat the process. After a couple of generations, I'm down to a single clone. Everyone in that population may show variation. That's all environmental. And my selection response stops. So when you select among clones, you run out of genetic variation almost immediately. If I select among outbred individuals, you have lots of genetic variation that continues to go. Why? Individuals are segregating alleles as heterozygotes, and that gives you a huge amounts of response. By the way, the 2018 book talks about all the population genetics of selection response in very considerable detail. I'm just kind of giving you an outline here. So that's the idea about the breeder's equation, response across generation change, namely the change in breeding values, equals the heritability, either narrow sense if I'm crossing the selected parents, or broad sense if I'm selfing the selected parents. And then S is the difference between the mean of the selected parents and the overall mean. Okay? So we might think that this selection differential S is a measure of the strength of selection. And let me show you that's not quite the case. So down below in the 3 panels, I've got 3 different scenarios and their associated S values. In panel A, my phenotypic variation is 4. I select the upper half, and the mean of that colored wedge to the right is 1.6. Panel number 2, the phenotypic mean is again 4. I select the upper 20%, and the mean of that little wedge, S, is now 2.8. And finally, in panel C, the variance is now much smaller, it's 1. I again select the upper 20%, and now my S is 1.4. So I simply gave you 1.6, 2.8, and 1.4. You'd say, well, the section is strongest in B, next strongest in A, and weakest in C. But what you see if you look at the percent saved, you see that B and C are the same and they're both bigger than A. And the reason that arises is S is actually a covariance. And a covariance scales with the variances. What we want is a measure that's independent of the variance. So for example, S down here, well, if I divide that by the square root of 4, S is 1.6 divided by 2 phenotypic standard deviations above the mean. If I take 2.8 divided by the square root of 4, I get 1.4. I take 1 divided by the square root of 1, I get 1. If I express this in terms of phenotypic standard deviations, B and C have the same response, and it's bigger than A. And we call that notion the selection intensity. We denote it by I. Sometimes it's a script I with the dot removed. Um, you can't actually write that font in PowerPoint. So while the Breeder's equation was originally expressed in terms of S, if you look at applications in the breeding literature, it's usually expressed in terms of the intensity I, which is the change in phenotypic standard deviations. We do this for two reasons. Number one is different schemes may have different phenotypic variances associated with them, and you get S inflated or deflated, not because you've made selection stronger or weaker, but rather your phenotypic variation changes. For example, I'm selecting at the average value of offspring versus a single individual. The second reason we do that is more interesting, and that is as a breeder, we usually talk about what fraction am I going to save. If the trait is normally distributed, which for most traits of interest are roughly distributed, if I tell you the fraction saved, I can exactly translate that into an intensity. So here's the expression that does that. This is the value of a normal distribution evaluated at this threshold point. There's the R code that will do that. So if you plug in, let's say 10%, this gives you the intensity. And here's what that table looks like. So if I select the upper 5%, my intensity is about 0.8. What does that mean? If I select the upper 0.5% of the population, the mean of that is 0.8 phenotypic standard deviations above the population mean. Select the upper 10%, the mean of that wedge is 1.8 phenotypic standard deviations above population mean. So the upper 5%, it's 2 standard deviations. So select the upper 0.5%, it's about 2.9. So what you see is diminishing returns as my intensity, as my fraction saved gets smaller and smaller and smaller, my intensity goes up, but it goes up in a very slow fashion. This is typically the range people select in from about 20% to about 5%. So intensities are about 1.4 to 2. So people use intensity for these two reasons. Number one, it directly translates fractions saved into into strength of selection. Number two, different schemes may have different phenotypic variances, and using intensities removes those effects. So one scheme may have a larger S value simply because it has a smaller variance associated with it, and the intensity is a way to remove those. So when you do that, you can take the Breeder's equation, and this is just simple algebra, and I can write it in this expression down here. Response equals intensity, square root of the heritability times square root of the additive variation. And this looks much less transparent than the Breeder's equation up here, but it's actually much, much more informative. H is the correlation between a phenotypic value and a breeding value. So the square of that, H squared, is how much fraction in the breeding value is predicted by the phenotypic value. So writing it this way, you can think of H as the accuracy. If I pick individuals based on their phenotype P, what does that tell me about their breeding value A? So I can write the Breeder's equation as intensity times accuracy times usable genetic variation. And again, at this point, it doesn't look quite that helpful. This allows us to generalize it. And this form, by the way, will tell us things like why genomic selection is very, very powerful. So let's take that one more step forward. Suppose in the typical Breeder's equation setting, I choose individuals based upon their phenotype. So I pick the tallest individuals or individuals with the most grain or with the most biomass and use those. Under more general settings, I can use some other way to pick individuals, for example, the mean of all your offspring, or you plus some weighted value of all your relatives. So now I replace your phenotype with some index which incorporates information from you and relatives. And I can now ask, how well does that index predict your breeding value? So if the index was simply your phenotype, this correlation is H. If I can make that correlation bigger than H, I get larger response. So to make this a little bit less abstract, let's look at a particular example. Again, the importance isn't the example or the details, but it's the logic. So one classic way that we, for example, do uh, breeding for dairy cattle is in dairy cattle, it's not very effective to select on females. Why? Female can't have, a female can have many offspring, but a male, be sure to mute, thank you. A male can have a very large number of offspring through artificial insemination. So almost all of dairy breeding works by picking those males with the highest breeding values. And a male with a high breeding value can have 1 million offspring through artificial insemination. So how do we do this? Because I can't score milk production in a bowl, but what I can do is progeny testing. And what progeny testing does is it takes a bowl and it crosses it to N random females and looks at mean milk production in their daughters. We're going to call that X. So what I want to know is, how does mean milk production in, let's say, 5 daughters, how well does that predict the breeding value of the father? Well, here's the expression that does it. It depends upon the number of daughters you look at and the heritability. If you plug this correlation here, which is this value here, so if I choose parents based upon looking at the average of N of their offspring, how well does that predict their breeding value? Well, there's the expression. And if you look, heritability comes in there, I pull out a heritability, I get an H. And so this term here is the advantage or disadvantage from using progeny testing. So I get I, H, sigma squared A. That would be the normal expression, choosing individuals just based upon their phenotype. This is the additional amount based upon choosing N of their offspring. If this is bigger than 1, I get a larger response. And this is bigger than 1 when the sample size is bigger than here. So for example, if the heritability is 10%, I've got to look at 4 offspring to have the mean of the offspring be a better predictor of the breeding value than the phenotype itself. If heritability is 50%, I've got to look at 7 offspring. Why? As heritability goes up, phenotype is a better predictor. You have to look at more and more offspring to compensate for that. We can then ask, what's the ratio of the response under progeny testing versus the response if I simply select individuals based upon their phenotype? You get this expression. And what you see here is that for really large N, this approach is 1 over H. So if the heritability is really small, you can get a much larger response by using progeny testing. As the heritability gets large, progeny testing is still effective, but the extra gain isn't that much. Now, the purpose of this example wasn't to walk through progeny testing. It was to show you how the breeder's equation allows you to replace selecting individuals based upon phenotype P here, predicting breeding value. That would give a correlation of H to much more general scenarios. So in particular, this is the correlation among between the breeding value of an individual and the mean of N of its offspring. So this generalized breeder's equation version allows us to look at much, much more general settings of choosing individuals to make the next generation independent of just their phenotype. I can add a bunch more information into that. So some immediate questions are, how can I improve accuracy, right? Single individuals, especially in plant situations, have pretty low accuracies. So if I pick a single individual, their narrow sense heritability may have a lot of environmental noise. Or if I pick a single clone, that is, I don't replicate, H squared can be pretty small. So how can I improve accuracy? Well, one way to improve accuracy, and instead of choosing the best individual, is to replicate. So if I got high G by E, instead of picking a single plant based upon the fact it's the best performer, what I might do is I might look at the mean of all its relatives over the entire population and then pick individuals that have the whose siblings have the highest mean performance. That's called SIB selection. And by looking over environments, that's a way to reduce G by E. So for example, instead of picking the single best corn plant over a series of fields, I might look at each corn plant and look at the mean of all its SIBs. And so for each corn plant, I have mean of all its SIBs over the entire population. Then I pick a corn plant who's has the highest SIB mean. That's a way to improve accuracy because I'm averaging out over G by E. And that's something called BLUP that we'll talk about a little bit later on. There's another way to do it. And this is an idea by Gardner in 61. And Gardner noticed with open pollinated populations of maize, if you simply pick the best 5% best plants in the field, you get very little response. Why? The field is very heterogeneous. And those 5% best may simply be those 5% that are in the best part of the field. So he suggested something called stratified mass selection. So you look at the field and you have some notion from past history about, well, in this field, the gradient runs east-west or north-south. So you block the field into a series of blocks or strata where within each block you expect the environments to be roughly equal. So instead of selecting the best 10 plants out of an entire field, you break the field into 5 blocks, and in each block select the best 2 plants. This idea about stratified mass selection is one way to try to control for the environment. So instead of comparing individuals over the entire field, you're comparing a smaller number of individuals within a section of the field where you expect the environment to be much more similar. And so that's one way to improve accuracy. Another way to improve accuracy is to just simply use SIBs and replicate SIBs over environments. And there's a whole bunch of schemes that do that. In fact, what I'll do is after class, I'll send on the chapter from Walsh and Lynch on SIB selection, which covers a whole bunch of these different schemes and tells you how to do that. I'll post that after lecture. So we're almost done with selection on the mean. Let's take that Breeder's equation. Remember, it was originally R equals H squared S, but now I can look at real world settings. And in real world settings, it takes time to measure males and females. If, for example, I've got a shrub like coffee or a tree, I could have an individual that has several generations of offspring while it's still alive. Likewise for animals. So what I now care about is not the response per generation, but the response per year. When I do that, that Breeder's equation now becomes this. So there's the heritability, there's the um, uh, there's the so if you notice, the reason it looks a little bit different here is I've broken this into, instead of I times sigma A, I've written this as H squared times sigma P. That basically is the same as saying H times sigma A. So that's the usable variation and the measure of the accuracy. This is the strength of selection on males and females based upon intensities. Again, these are percentage. And now these are the generation intervals. What's the average age of a mom? What's the average age of a dad? So for example, just quickly, suppose I've got a scenario where I've got 90 dads and 60 of them have offspring in year 2 and 30 of them have offspring in year 3. So the average age of a dad is 2 times 60 divided by 90 plus 3 times 30 divided by 90, or about 2.3. In females, the generation average is longer. Why? Because if they're going to give multiple births, you've got to wait several years. So now I've got 11,040. So what fraction of females are 2 years old? It's 400 divided by 1140. Multiply that by 2. Same for 3, 4, and 5. So this down here then, that's the average age of a dad when he has offspring. That's the age of a mom when she has offspring. You notice that moms on average are older. And if you look about older, that drives us down. So when generation intervals get longer, response gets smaller. So one critical thing that genomic selection does is it allows you to evaluate individuals very early on. I might be able to look at a two seed stage of a seed. On that two stage, predict what the trait value is going to be with low accuracy, but still a prediction. And by doing so, I can greatly reduce the generation interval at the drawback of lower accuracy. But if my generation interval is reduced by 10 and my accuracy is reduced by 4, I get a larger response. Likewise, what genomic selection does is allows me to have much stronger selection. If I can score an individual when they're 2 days old, I may be able to score 10,000 individuals. If I've got to wait until they're a year old, I can maybe only score 100. So genomic selection doesn't necessarily work by improving accuracy. It mainly works by shortening the generation intervals, often really dramatically, and also greatly increasing the strength of selection. Again, we'll talk about that later on when Nelson and other people talk about genomic selection. So this is the generalized breeder's equation, and it shows you the trade-off between generation length, selection intensity, and accuracy. These are things, that component, and that component are things you all have control as a breeder. The problem is they're trade-offs. Think about progeny testing. Progeny testing, I replace the mean milk production in your offspring by your phenotype. That gives me a higher prediction, gives me a value here bigger than H, but it takes more time to score. Does that trade-off work? Well, chapter 13 shows you the numbers and whether that trade-off works or not. So as a breeder, you can control the intensity, the accuracy, and the generation intervals, and there's often conflicts between those, right? So these are all components you can breed, and then there are conflicts in between those. So let me stop now and ask if people have any questions about response in the mean. Let me give you the key points, first of all. Most important key point, follow the general structure of the lecture, not the details. You can then go back and read chapter 13, which covers all the details and much more, gives you lots of worked examples. So the main features we talked about so far is you can measure a within generation change. That only translates into a change in the offspring if that within generation change in the phenotypes of the parents results in a within generation change in their breeding values. And the connection there is the heritability. If a trait has a high heritability, your phenotype is a good predictor of your breeding values. If the trait has a low heritability, it's not. And the connection is H, the square root of heritability, is the correlation between your phenotype and your predicted value. Then we talked about selection intensity. It's basically rescaling S to measure it in terms of phenotypic standard deviations. The strength of that is if I give you a fraction of the population saved, you can translate that into an intensity. Then we talked about how you can decompose the breeder's equation into selection on males and females, generation intervals, and accuracy. So those are the key ideas from the first part of that lecture. And again, as before, I'll look at the chat first to see if there are questions in there. Come on, chat, you can come up. The chat doesn't want to come up here. There we are. There's the chat. Previous lecture notes, lecture slides for today. Yeah, so if people have questions, just go ahead and raise your hand on the changes in mean or type it in the chat. After a minute or two, we'll go back and talk about predicting changes in the variances. That's the so-called Bulmer effect. Again, I remind you that today's lecture is going to be somewhat short. We'll be done in an hour. Tomorrow's lecture, the lecture itself will probably be right around an hour with questions after. And Thursday will be around maybe 65 to 70 minutes. So a little bit longer on Thursday. It may be quicker, but just prepare for potentially a little bit longer. So any questions? Just go ahead and raise your hand or type them into chat. See a whole bunch of people here. Okay. Okay, well, if not, give me a thumbs up if we should move on. Just use that reaction bar down at the bottom. Like, here's me giving me a thumbs up. There's a thumbs up. Okay. Fraud, you have a question. Go ahead. Faud, F-A-U-D, you have your hand up for a question, or is that just a thumbs up you hit by mistake?
I have a question.
Please.
Okay. Are you hearing me?
We can. You can go
Uh-huh.
ahead.
Okay. Just it was an interesting lecture. Just I wanted to just when we are dealing with traits that are severely affected by biotic stress, or specifically, I'm focusing on, for instance, parasitic weeds. Field patch can completely mask a plant through genetical potential. So in that case, if environmental noise is that much extreme, is there a way that can be stratified mass selection still hold up? Or
Yes.
are we forced to pivot entirely to family-based replication or genomic selection?
So that's a great question. So if your trait is expressed serially, so you get over time, one way is to treat it as a series of correlated traits. We'll talk about that on Thursday. But the other way is family selection is generally a pretty good, robust approach that allows you to handle settings. And most plant breeding situation is typically family because you have a lot of G by E. So if you've got a pathogen problem, you're crazy to measure a plant in just a single plot because that plot could have low or high pathogens. You want to replicate it over a bunch of environments. So most plant breeding situations is some form of family-based selection. And that's how you handle really noisy traits.
Okay, thank you very much.
Next question. Yeah, Kuldeep, you have a question? Kuldeep, again, apologies if I mispronounced it. If you have your hand up, go ahead and speak up.
Hello.
Yes,
Yeah,
hi, go ahead.
Bruce. Yeah. I just tried to type my question on chat box
Okay.
also, but
Oh,
basically I
there it
just
is.
want to
Yep.
know, yeah, like more into like when we want to go for elite by
Yep.
elite cross combination. So this selection intensity and its impact on a mean and the variance. I just want to explore like as a breeder how it's going to impact and what could be the strategy where you can improve in a better way. So it might be
Yeah,
a
so
yeah.
yeah,
I
so
hope I
great question. Go ahead.
yeah, please you can go ahead. Thank you.
Yes, so one of the we've talked this lecture is about mass selection where I'm taking a bunch of a population and sampling them and choosing the best parents. The slightly different question is if you're going to make elite lines to cross, where now you choose a single parent to cross to a single elite line. And there are a variety of ways of doing that. For example, you have heterotic groups, you want to choose individuals from different heterotic patterns. But as we'll talk about with genomic selection, you can use BLUP to basically use all the information to find those best parents that you want to cross. Um, and in that case, the intensity, it's a little bit tricky to classify it. The easiest way is simply to use genomic selection because that will predict what the response is going to be. Are you able to use quantitative genetics to breeds prone to mastosis? Yes, you can. So there's a lot of work done in dairy cattle breeding on how you select for mastosis. The problem with that is it's a binary trait, 0, 1. The same logic holds, and chapter 13 gives you some examples of how you work through that. Okay? Innocence, go ahead. Not so. Go ahead. Not so innocent.
Yeah, my questions are a little bit practical. When we talk about of course, genetic gain, it is a function of how we conduct our experimental trials and obtain quality data. Now, when we talk about family selection, let's imagine we are running early generational trials. Maybe
Right.
crosses are being generated with different family sizes, and then you have to go maybe for logistic reasons, you can't replicate them. We could say we go for augmented design, where maybe we replicate only the checks. Now, the planting structure of this experiment would be, one can say, I'll be planting my designed, maybe per block, will be family-based. Each family, for example, if I have 30 individuals per family, I plant them in that order. Another way of doing it is like, oh, I could just randomize it and plant them randomly across the field. So in unreplicated trial, what is your take on dealing with planting families together or trying to spread them across within the experimental field in
Yeah, that's
terms
a
of obtaining really a better outlook of how they'll perform? Is it better to put them together and observe how they perform, or we can spread them across? If you spread them across, since they are just single plants, are the observations reliable, or they could be influenced by maybe field structures and all that?
Sure. Yeah, so and again, just to remind people, as was noted in the thing. So this is the first half lecture. We're going to go to the second half in a second, but let me answer innocent or not so innocent question. Um, and that is when you've got complicated designs, like when you replicate families. What you typically do is you use the design then to remove the effects of the field, and then you either have a blue estimate of the family value if you treat them as fixed effects, or a BLUP estimate if you treat them as random effects. The advantage of BLUP, again, we'll talk about this later, is you can borrow information from all your relatives. And typically these people design where people have planned them over, what you do is you pick individuals based upon either a blue estimate of line value or a BLUP estimate of line value. I'm a fan of using BLUPs because you can borrow information, because you can use markers. If you've got marker data, you can use markers to get relatives. But the short answer is the way you handle more complicated designs is you extract from the design either a blue, an ordinary least squares estimate, or a BLUP, a predicted value from your design for the genotypic value, and then use those to make your crosses. Okay, let's move on now. Let me finish up here. Let's now then go to the second aspect I want to talk about. And I want to remind people that there's aspects about this which are a little bit technical. Don't get hung up on those. Just follow the general flow. This stuff is all covered in chapter 16. And this is something called the Bulmer effect. And this is for predicting changes in the variance. And to do that, we need to talk a little bit of technical stuff. Don't worry too much about it. If you're really into this, the 2018 book has like 200 pages on this. But when I change selection changes the variance of a trait. When I change the variance, I change the additive variance. When I change the additive variance, I change the heritability. So heritability is changing. So R equals H squared S is a predictor of one generation. Now, the heritability can change from two features. One we typically ignore, and that is selection changes allele frequencies, but for most alleles, the effects are small. That change is tiny. It takes dozens of generations for that to come up. The other one is something called linkage disequilibrium, and that can have a larger effect, but it's actually quite predictable. And linkage disequilibrium simply means that alleles at different loci turn out to be correlated. Again, don't think about this too much. Just follow the general logic on here. Chapter 16 talks about this in more detail. But the idea is, suppose I pick out a random pollen grain. And in that random pollen grain, I've got allele big A at one locus and allele big B at a second locus. If in that sample, the frequency of pollen grains containing a big A, big B gamete is the same as the frequency of A in that sample times the frequency of B, we have what's called linkage equilibrium. If I have a big A, big B is no more or no less likely to be found compared to a random sample from that population. Positive linkage disequilibrium, called positive LD, is when that combination is more frequent than you expect by chance. So A and B are positively correlated. When you get a big A, you tend to also get a big B. Negative disequilibrium is when the frequency of that combination is less than you expect by chance. It means A and B are negatively correlated. When you get a big A, you're less likely to get a big B. And selection can generate these combinations fairly quickly. And so if you look at what additive variation looks like with LD, this is the sum of all the breeding values, summing up all the effects. If I ignore these correlations for a second, this is just what's called the genic variation. This is the part of the genetic variation that changes as allele frequencies change. This is due to correlation among alleles at different loci. Sum together, that's the disequilibrium. And the key is selection can generate D in a very predictable manner. And when D is changed, it's usually negative, and negative D causes selection to decrease. And again, I'll tie all these pieces together. Again, don't worry too much about the details. You can read through it in chapter 16. The key idea is that when you select, you tend to generate correlations between alleles at different loci. That generates a negative D, and that means your heritability is decreased, but it's decreased in a very predictable fashion. So here's the idea. Here is my trait variance. It's environmental variance plus the genetic. Genetic has dominant plus additive. Well, this additive down here, go back here, has a part that doesn't change. That's allele frequencies. We're going to assume those don't change. It's got another part that does. That's the disequilibrium. So this is the variation in the next generation. So the heritability is simply the initial heritability plus the disequilibrium. The phenotypic variation is the initial phenotypic variance plus the disequilibrium. So if I have some way to compute how D changes, I can tell you how the heritability is going to change. And it turns out there's a very simple way it does that, and this is what's called the Bulmer equation. So there are two ways D changes. Number one, each generation, the initial D value that's passed on is cut in half. Why? We assume these are unlinked loci. So when you stop selection, disequilibrium declines by a half each generation. However, selection generates disequilibrium. So what type of D is generated by selection? Here are three panels, and that circle, imagine that there's the allelic effects. So X and Y are simply two loci, and that circle is the whole distribution of effects. So you could have an allele with a big effect on X and a little effect on Y, or a big effect on Y and little effect on X, etc. So the circles mean that there's no correlation between those effects before selection. If I have directional selection, it means the allelic effects are bigger than some number. What I end up getting is that shaded slice here. In that shaded slice, the initial circle, there was no correlation. In this shaded slice, when I tend to be big for X, I tend to be small for Y. When I'm big for Y, I'm small for X. So directional selection generates a negative correlation between the allelic effects. Same for stabilizing selection, that is selection to decrease the mean. The only one that generates positive D, so if you look at these two wedges and fit the curve through there, you get a positive line. That's disruptive, which is unusual. It means you're selecting for individuals that depart from the mean, not that stabilize around the mean. So most selection generates negative D. If I choose individuals to be larger, I generally find that alleles that are larger at one locus tend to be negatively correlated with alleles which are larger at a second locus. And that generates negative D. So here's my parent-offspring regression. If I then plug in the mean of the variance among the selected parents, again, don't worry about the details. You can look all those in the chapter sign. This measure here is the change in variance in the parents. So when I choose parents, the variance among those parents tends to be smaller than the variance of the entire population. That's that disequilibrium term. That disequilibrium term there turns out to be a function of the square of the heritability. And from that, then, I can predict. So the disequilibrium is the original value divided by 2. That's the decay from recombination. And then the part from selection. So this is the reduction in variance in the parent. So I have a whole room full of people. I measure a variance. Then I choose a small subset. If I choose, let's say, the tallest, the variance among those tallest is going to be smaller than the overall variance. This term here would be the variance among the tallest minus the overall variance. That would be negative. This is the part of D that's generated by selection. So the previous D is cut in half by recombination, but it's generated by selection. And it turns out that's the equation that tells you how disequilibrium changes. It turns out it's pretty easy to quantify this under a lot of scenarios. And this expression here for D is called the Bulmer equation. And just kind of jumping ahead here, in particular, again, I'm doing this to show you to not worry about the details from the notes. If I have scenarios where the mean of the selected parents is some fraction of the overall mean, I've got very nice expressions that allow you to quantify that. And in particular, with directional selection, where I save the upper, let's say 5%, this coefficient k in terms of how it change, I can express that in terms of intensity. So the bottom line is, if I have directional selection, that gives me a measure of how much disequilibrium reduces the variance and allows me to use the Bolmer equation. So in particular, what you can show is that, going back a second here, so at equilibrium, that's the disequilibrium at disequilibrium. So these things here, square the heritability and change in variance, those all change with the disequilibrium. So Bolmer gives this example. So he looks at egg weights in ducks. And you have a whole bunch of eggs in ducks, and some don't hatch. You measure the weight of all the duck eggs, and then measure the weight of the duck eggs that hatch, and the mean doesn't change at all. What you notice, though, is the variance before hatching is about 53. The variance among those due to hatch is about 44. Heritability was about 0.6. So the equilibrium disequilibrium value is 0.6 squared, and this here is the mean of the variance of all the eggs, the variance of the eggs that hatch. So your disequilibrium here is about minus 3.2. And what that says, you run through the numbers, it says that you decrease the variation from about 35 to about 32, and the heritability should usually be about, before selection is 0.62, it goes down to 0.6. The effect is small here, but it can be more dramatic in other cases, as we'll give an example here. So let's finish off by going through the following example. So this is the reduction. That's that kappa that you plug into the Breeder's equation, the Bolmer equation. That depends upon the intensity. This is also a function of the strength of selection. Again, just worry about the rough details. Chapter 16 gives you all the information in here. Suppose I select the upper 20% of the population. My heritability is 50%, and my starting variance phenotypically is 100. So if I select the upper 20%, that gives me an intensity of 1.4. There's that z value that I use. So my reduction in variation is about 0.8. So if I select the upper 20% of the population, the variance in those individuals is going to only be about 20% of the variance of the entire population. So I can then plug all those numbers into the Bolmer equation. That is 100 times 0.5. That's the added variation before disequilibrium. That's the phenotypic variation before disequilibrium. This is kappa over 2. And this is the equation that allows you to iterate. So the only thing here that changes is d. So it's an Excel spreadsheet. So at generation 0, you've got 50 plus 0 squared divided by 100 plus 0. So you basically, and then you basically then have 50 squared divided by 100, and then minus 0.39. And this term here is 0. So in the first generation of selection, here's your initial d, here's your added variance, here's heritability. Now, next generation, you generate a d of minus 0.9. That changes your added variance from this plus d, which gives you a variance of 40. Your heritability goes from 50 over 100 to 50 minus 0.9 divided by 100 minus 0.9. So it now goes down to 45. In the next generation, you just plug this number back in here for d of 1, iterate, you get disequilibrium of minus 12. Your added variation goes down to 38. Your heritability goes down to 0.3. You plug that in, and as you generate, you see that you very quickly, after a couple of generations, get your final value. So after a few generations, you start out with a heritability of 0.5. You now end up with a heritability of about 0.43. It's a big effect. And this all arises because selection is generating negative correlations among favorable alleles. And this is called the Bulmer effect, and it allows you to track that. So in particular, to close off, there's the Breeder's equation. And in the Breeder's equation, what is changing is the heritability. So my initial response in the previous example was 7 per generation. If you plug in the equilibrium values here, you get the response goes from about 7 to about 5.6. So the Bulmer effect, and the key about the Bulmer effect is what we're simply doing is trying to predict how the variance is going to change. And usually over the first couple of generations, the added variance and therefore the heritability drops maybe 20, 30% and then stabilizes from that. That is entirely predictable. And the Bulmer equation allows you to predict that. So the Breeder's equation will overpredict the response in one generation. Now, if you're worried about the details, when you use genomic selection, this is all baked into the expressions for you. So in this lecture on single trait selection, I've talked about number one, changes in mean, that's a Breeder's equation. Number two, changes in the variance from selection generating disequilibrium, correlations among alleles, that's called the Bulmer equation. So the Bulmer equation isn't talked about as much, but you see it a lot in animal breeding because it is real and it does reduce your predicted responses by 20 to 30%. Okay. And that's the end of the lecture. Again, what I really want to stress is the Bulmer effect is a little bit technical. Don't get hung up on the technical things. What's the key idea? When you select, you get an immediate reduction in the variance. That reduction, as heritability gets larger, the reduction gets larger. As the strength of selection gets larger, that reduction gets larger. It comes to an equilibrium value fairly quickly, 2 to 3 to 4 generations. If that's the main thing you take away from that, that's 90%. The details about how the equations work and everything, those you can look up on the notes for chapter 16. Okay, so we talked about changes in mean, Breeder's equations, most people know that. Then we talked about the Bulmer equation, changes in variances. So remind you before we get to questions, tomorrow we'll give an overview of matrices. I've sent the notes, those may or may not be posted yet. And that's going to be, the lecture itself, there will be about 60 minutes, a little bit longer than this lecture. And then on Thursday, I'm going to talk about multiple trait and index selection. I've got about 70 slides, which means roughly 65 to 70 minutes. It might go a little bit quicker. So just be aware and plan ahead that my last lecture on Thursday is going to be just slightly longer than the lecture today and the lecture on Tuesday. Okay? With that, I'll see if there's any, and by the way, one more thing too, is I will then forward on to be post the chapter on selection based upon family or SIB information. I'll post that. I think it's chapter 22 or 23, but I will send that immediately on to be posted on the course website. And you can find it there for those working with selection on families. A lot of bookkeeping there, a lot of different schemes, but I'll send that chapter for you. So, general questions.
So there was a question that came through a little bit earlier from Halima. Halima, if you're still on the call, do you want to go ahead and just speak to your question? Just checking if Halima is still on the call. We might have lost them.
Yes, and
Yeah.
by the way, when you're asking your question, if you want to, please use video. It makes it a bit more personal. You don't have to, but if you want to use video, it makes it a bit more personal as well. While we're
Okay,
waiting
well,
for
it might be that Halima has dropped off. I'm not seeing her in the contact
okay.
list, so you're welcome to continue, Prof. Sure.
Paul, you have a question. Paul, go ahead.
Thank you. Excellent presentation. It helps me understand some things much better. But on the Bulmer equation and the heritability calculation, as the additive variance reduces, doesn't the phenotypic variance also reduce?
Yes, and that's a good point. Let me pull that back up here. That's actually taken into account here. Let me pull this back up here. So when you look at that expression down here, let's go back to the Ulmer equation, get back here. So it's down here. So there's the phenotypic variance. So I go back and forth between Z and P. Apologies. Z is used in the evolution literature. P is used in the breeding literature. So there's reduction up here in the additive variance. There's reduction in the phenotypic variance. So it exactly accounts for that, that when one goes down, the other goes down. The reason the heritability goes down is the reduction in here is larger than reduction in there because this has other components, so that mitigates it to some extent. But yeah, so it does account for the phenotypic variance also changing as the additive variance changes. Yeah, good point to clarify that.
Thank you.
Any other questions? Or are people still recovering from? I'm not sure in Africa what time the game ended. Maybe like 4 in the morning or something.
Most
Okay.
of the games are in the middle of the night, so it's definitely not the best hour.
Yes.
Any other questions? You are still welcome to raise your hand or type in the chat. We can pause for just a second or so, give folks some
And
time.
let me remind you as well that my email's at the beginning of each slide. So if you have a particular question and want to email me, please feel free to go ahead and do that. And we can also, by the way, always Zoom. If you have a more elaborate question, email me and maybe we can set up a time to Zoom about that as well. Happy to do that as well. Okay, any other questions?
I don't think there are any
No,
other
applause.
questions.
Okay. So if not, then tomorrow, same time, same place, I'm going to review
Hello,
matrices. Yes, go ahead.
hello,
Desmond, go ahead.
hello.
Desmond, go ahead. Oh, no, not
Come
Desmond.
again, maybe I'm come
Raise your
again.
hand so I can see
I've
this.
got a network break.
Just put your hand
Sorry,
up.
I've got network breakdown. So I have network breakdown. So can you kindly come back? Can you kind
Sorry,
Talon,
not sure what happened there.
Talon, if that was you asking a question, go ahead and type it in. So if you want to ask a question, use the raise your hand button at the React. What that does is it pushes your picture to the right of the queue, and I can actually see who's asking the question. Right now, you're on 3 separate pages. So just use that. It's under React, the heart, and there's a hand raised. Just use that if you want to ask a question. It moves you right to the front of the queue where I can actually see who you are. And don't be bashful. No?
There's one more question that has come through on the chat. Debo Prio, if you want to ask it out live, you're more than welcome to, but it's quite clear on the chat. Would you like me to read it, Prof?
Also, I put my email on the chat because someone asks what my email was. So it's down there as well. Yes, please share your email. Oh, okay, there we are. And that was Paul's question. I wanted to ask if genotypic values remain the same in a population, does the add? That's a great question. So the genotypic values stay the same, but imagine it this way. I've got a room of a whole bunch of different genotypic values. I then pick out a sample of those. The genotypic values in the sample have not changed, but the variance of that set of genotypic values is different from the variance of the overall set. So the additive values have changed in that, sorry, the additive variance has changed because that set is a smaller sample of the entire population. So by chance, it could be the variance hasn't changed, but in most situations, the variance has changed. In particular, the breeding values tend to be larger in that set of parents than the overall population. In an overall population, the mean breeding value is 0. If I take a subset of that population based upon large breeding values, the mean of that population is that subset is going to be different from 0, and the variance is going to be smaller than the variance of the breeding values over the entire population. And that variance that we measure is the additive variance. So genotypics don't change, but my population sample gets smaller, and therefore the variance, the spread among those individuals which don't change gets smaller. That's the basic idea. Okay. Someone's microphone is not working, but go ahead and type it in. And I see that you did type it in. Okay, any other questions down there? Sorry for doing this, but I've got very poor eyesight and the fonts are really tiny on here.
So there's no other questions that I can see on the chat at the moment, and there are also no hands raised. So I think we are okay. You have answered their question.
Okay, fantastic. I will see whoever shows up tomorrow. By the way, even if you had matrices before, it's good to get a review on that because we're going to be using them on Wednesday when we talk about multiple trait selection. And Wednesday afternoon, I'm headed right here. So after the lecture, I'm headed off to the mountains. Okay, you guys have a great day, or a great evening, I should say. It's 6 a.m. in the morning here, so good luck. Cheers, everyone.
Thank you so much.
Bye bye.
Bye-bye.